HPV using cell cultures?
HPV is a sexually transmitted infection, and it's based from DNA, and it results in like genital and oral warts. And there's more than 40 different types of HPV, and some are more extreme than others, and it can lead to cervical cancer, so it is a carcinogen.
So what we did for our experiment is we had we had 10 different petri dishes in total. 5 of them were used as controls, so they didn't have any HPV infection, but the other 5 had varying amounts of it, from anywhere from 5 to 500 micrograms. And these were checked every week with a scratch test to check for cancer. And each week we compared one of the infected to one of the controls to see whether it had cancer or not.
Wait, go back. 5 different petri dishes of what?
Of cell cultures. Cells. Cervical cells. So this is our table of results, and we check them every week. And as you can see, the trial with the highest dose of HPV, they all read positive every week. So as the dose level increases, the amount of cancer increases, or the presence of cancer increases. There were a few controls that had it, but this could be due to other environmental factors or something else that we are not aware of. But
can you explain what those plus and minus signs mean? Because they don't.
A plus means that it read positive for cancer after a scratch test, and a minus means there was no cancer after a scratch test. So you scratch them and you see how quickly the cells fill in the open space, and you determine if they have cancer or not.
In conclusion, we found that HPV is a carcinogen, and as the exposure of HPV increased, so did the risk of contracting cancer. And also as the time increased, so did the amount of cells that got cancer.
And to take this farther, we would use mice so that we saw how HPV affected an entire organism and not just a couple of cells. And I think we'd keep the time and dosage, size of HPV as the variables, but we might use smaller increments, because if you go back to the table, from the 4th trial 4 to trial 5, we had a 200 microgram increase, and we just wanted to make sure that the reason that it went from just 2 getting cancer to 5 was not, I don't know, so we had more trials in between the smaller increases.
Any questions?
Okay, let's go back to those results first. So you concluded that from these results, and I want you guys to look at these results a little bit more in depth. They concluded that HPV is a known cause of cancer based on these results. I want to caution you guys a little bit about drawing conclusions from one experiment. So just looking at this experiment, is that a conclusion that you feel totally confident in?
No.
Yeah, I agree with you. I don't feel totally confident with that conclusion either. Why not? What are some of the reasons we would not necessarily say? I mean, I understand why you said yes, it's a carcinogen from this, but I also want to caution you from making a conclusion not definitive. You know what I'm saying? So, why would I caution you there? Anybody have some ideas, Ben?
Because
Well, on the 3rd or the 2nd trial, that there's only 3 that were positive, and then on the next one there's only 2, so it goes down.
Yep.
like in week 3 on trial 2, it is positive, and then week 4 it's not, and week 5 it is positive.
That was because we used, for each trial there were 5 different dishes with the same trialing that we did at the same time, because we didn't want to do a scratch test more than once.
Okay.
So the one on 4 is a different one that was
Oh.
looked at than the one on the 3rd
Alright.
week.
Yeah, it's not the same cells that went non-cancerous after they were cancerous. That didn't happen. But that's a good question. But they still saw that that's some variability, right? And if you had a very, very strong connection, you wouldn't expect that result, right? Okay. Anything else that makes you feel like, yep?
Since the control tested positive a couple times, I would feel a little uncomfortable if the experiment's almost compromised, since that means they do have some hidden variables that they can't exactly control in their experimental design. So that almost compromises the experiment.
an interesting point too. So I agree with these guys that the next logical step would be to look at some organismal levels, but I would even do some, and I told them that they had to do something different. But I would even look more in depth at this cell culture level, right? Because we still have some very big unanswered questions at this level, even before we go on to an organismal animal study level, right? You guys agree with that? Okay, very nice job, you guys. Very